Kia ora tātou, welcome to this next video where we're talking about another process that occurs during meiosis called crossing over. And I'll just give a quick apology for my last video. I said that it was called random assortment a few times, but independent assortment would be a more correct name. But as I say, the names, you won't get marked for the names that you used, so that's all fine. So variation from crossing over. So what happens during meiosis, and so we went over this just before, that we have the chromosomes lined up, so we can have a paternal chromosome, let's do it like this, paternal chromosome and a maternal chromosome. In this case I'm just going to do one pair of chromosomes because that's all we need to focus in on. So if we were to zoom in on these two chromosomes, what can happen during meiosis is that we get the chromosomes, and this is a very apt name, they cross over. So you can see them crossing over there. And then what happens is they actually swap some genetic material. So this part of this chromosome ends up attaching to that chromosome, and this part ends up attaching to that chromosome. So let's draw that so we can make it a little bit clearer. We'll go zoom in on that again. We'll go this way. So we've got the blue part of the other chromosome is attached to the black chromosome there, and then the blue chromosome is going to get a little bit of that black chromosome with it. So we get the blue going like that. So we can see we've swapped sections of DNA in homologous chromosomes. So in this case, what we would have if we had these two chromosomes forming gametes is that we would get one gamete would get that chromosome, another gamete would get this chromosome, another gamete would get this chromosome, and another gamete would get this chromosome. So we'd actually end up from this with 4 different gametes that all have unique DNA. So they would look like just a straight one black chromosome, then there'd be one with a little bit of blue on it, and then there'd be a blue one with a little bit of black on it, and then there'd be a straight blue chromosome. So that's crossing over. That's another way that variation can be introduced in meiosis. So we get genetically unique gametes from this. So again, it's not super important to be able to explain this in depth in an exam, but we just need to be able to talk about how we do get variation in the gametes. So here's a potential answer that you could use if it came up. So during meiosis, homologous chromosomes can swap genetic material. So we don't actually need to use the name crossing over, we can just say they swap genetic material. This results in gametes having a unique combination, and we can say a unique combination of alleles, to be a bit more specific. But if you said a unique combination of genes or anything along those lines to show that the gametes are unique, that's all you need. So that's the next, that's the 2 forms of variation that can come in during meiosis. In the next video we'll look at variation from fertilization.